and welcome to this lesson for higher biology, which is on proteins and phenotype. So up until now we've looked at the processes by which proteins are actually produced, that being transcription and translation, and how we get from a DNA molecule to producing a polypeptide chain. What we're going to look at today is how those proteins, once they're produced, actually function within a cell, what different roles they might have, and how that relates to the phenotype of the organism. Moving on, we will start looking at cellular differentiation, how those proteins that are being produced can influence the cell to produce different roles within the organism. So what you need to be able to do is explain how there can be more proteins than there are genes. You need to describe how folding of a protein can change its shape. You need to explain how the shape of a protein can affect an organism's phenotype. So first of all, looking at how we can have more proteins than we have genes. So the genome of an organism is roughly how many genes that we have present in our DNA. So how many small sections of DNA code for a protein. And in humans this is around 20 to 25,000 genes. What we have is also something called a transcriptome, and that is really what's produced by the process of transcription. So how we get from those genes are transcribed, the DNA is transcribed into mRNA, and the mRNAs are transcript. Remember, we've got our mature transcript, and those transcripts are referred to as the transcriptome. So as you can see here, we've got 100,000 transcripts from around 25,000 genes. So how do we get that much bigger number? There's a couple of different ways. We can have something called alternative promoters, which we're not going to discuss, alternative splicing, or mRNA editing. From the transcriptome, we can have 100,000 transcripts. We can wind up with over 1 million proteins, referring to something called the proteome. And that's achieved through something called post-translational modifications. So to begin with, looking at how we increase our genome to the transcriptome, looking at alternative splicing. If this sequence of letters was the sequence available on our DNA strand, for example, or a long chain of A's, G's, C's and T's, that can be put together in a few different ways. So if we were just to take out some of the sections, removing the red bits, if we're going from DNA to mRNA, splicing, removing the bits we don't need, keeping the useful bits, so we've removed our introns, the red bits have gone, kept the exons, which are the useful bits, and that's given us our final message, our final mature transcript, and that's going to be taken off and translated. Alternative splicing talks about the fact that not only are we getting rid of those introns and putting the exons together, but if you're giving it a couple of different options. So if we take the same sequence, and this time we have a few different introns and exons. So we've taken our DNA molecule, we've created our mRNA molecule, and we're going to remove those introns and keep the exons. So therefore we wind up with a much shorter and different mRNA transcript from the same gene. So what's important about that is that both of the sequences to begin with were the same. The mRNA was just spliced in a different fashion. So different exons and introns were used or not used to give us a different transcript, which would then correspond and give us a different amino acid sequence when it was eventually translated, and therefore a different protein. So we can wind up with very different mRNA transcripts from the same gene in this process called alternative splicing. And that's what this would look like overall. So if we have our primary RNA transcript up here, then if we're going to take away all of those blue bits there, which are our introns, leaving behind our exons, however they can be put together in different fashions. Over here this mRNA molecule has wound up with 5 of the introns, it's missed out number 3. This one's got 4 of them, it's missed out number 2 and number 4. This one's got another 5, it's missed out number 2. And in a similar way, so just literally with 6 different exons, there's so many different combinations that you could potentially come up with. So that's one of the ways that we can really increase the number of proteins produced from a small amount of DNA, is by putting them together in different fashions using those exons in this process called alternative splicing. Once the polypeptide chain is produced, we can fold that protein into different ways, and that allows a different structure to be produced. So if this is our original polypeptide chain, it's referred to as the primary structure. Hydrogen bonds can form between amino acids, so that one could become bonded to that one, for example, and in doing so, it forms what we call the secondary structure of amino acids. And this takes the form of either an alpha helix or a beta sheet. You don't need to know those terms, just that we can fold this protein, and initially is folded into its secondary structure. These secondary structures can fold again to form a tertiary structure, and some proteins would then join together. So this one here would form this yellow blob over here on this one, and this would be the quaternary structure. So this particular protein would be made up of 4 different polypeptides all stuck together. As a result of this different type of folding, we can have proteins that have different shapes and different structures. To begin with, we have fibrous structures where our polypeptides are arranged in ropes, very similar to a fibre, winding in and around each other, and those are used in cells for support, and often in connective tissues such as keratin or collagen, which are around and near your muscles. For example, keratin is what your hair and your nails are made up of. A globular protein is a little bit different. That's where those chains are wrapped around and in complex shapes, so they've been folded up, kind of blobby, but they are folded in a specific fashion. And that's when a particular biochemical shape is really important. So if it's an antibody or an enzyme or a hormone, something that needs to act or respond to another part of the cell somewhere else, it needs to have a particular shape so it can carry out that interaction. A conjugated protein is really just a protein which has been attached to something else. So if this is our hemoglobin protein, we can see we've got a few different polypeptides stuck together. It's also been labelled with a couple of the different secondary structures, but you guys don't need to worry about that. What's particularly important is the fact that it's also been attached to these little molecules here, which are referred to as heme molecules. Now, they're not made of protein, and what they're doing is they help those molecules to bind oxygen. So hemes is available to bind lots of oxygen, and that's attached to hemoglobin in the red blood cells, and of course the red blood cells' job is to bind oxygen. Finally, looking at what would happen if those proteins are changed. So the final expression of the proteins, what's actually produced, is going to decide what the cell can do overall. So their shape is determined by the mRNA, as it determines the particular order of the amino acids, and the amino acids determine how they're going to be folded. As a result, that folding is going to give them a particular role within the cell and within the organism itself. The production of those proteins therefore leads to what we call the phenotype, so the physical characteristics of an organism. And that can be something that you can see, or it could be something internal. It could be an enzyme which is working or not working. So when the protein is changed, that happens as a result of a mutation in the DNA somewhere. So the DNA itself has been changed, which affects our overall mRNA, which affects the shape and size of the protein. And in this case, that might just be that this particular one gives us a slightly different colour, so you would see that as a physical characteristic. That's one that you would actually be able to see the difference between brown eyes and green eyes. So the characteristics produced by the proteins are the phenotype, and that phenotype could also be influenced by environmental factors. So you should know by now that there can be more proteins than there are genes, because primarily of the process of alternative mRNA splicing, so putting together different exons to produce our mature mRNA transcript. That folding of a protein can cause it to be either fibrous or globular, or it may become a conjugated protein and join up with something else. And that the shape of a protein can affect an organism's phenotype, because the overall result of that protein may produce something else within that organism. a different physical characteristic that you could see, such as the colour of eyes, or it may be an internal change. It might be something like an enzyme. Obviously, the shape of a protein. Enzymes are made of proteins, so the shape of an enzyme is really important. It needs to have the correct active site to work on whatever it needs to work on, whether that's a degradation or a synthesis reaction. And what it's going to be able to do is break that down depending on its shape. If the protein's been mutated, then it may not be able to carry out that reaction anymore. And again, that would be an internal change, it's not necessarily one that you could see. So that might affect an organism's phenotype that way. As ever, feel free to ask any questions about any of this as we go over it later on.